Hello and welcome to the Galien Foundation Week of Innovation webinars. This is day 2, focused on biotech, and this is our second webinar of the day. Because the mission of the Galien Foundation is innovation to improve the human condition, we developed this webinar to showcase the innovation of our nominees in greater detail. This particular webinar is devoted to the 8 nominees in cancer, rare diseases, and vaccines. And the nominees are Merck & Co. for Ervebo, a vaccine for Ebola virus, Sanofi for Cablivi, a nanodrug that treats ATTP, Dompe Pharmaceuticals for Oxervate, a recombinant nerve growth factor to treat neurotrophic keratitis, Takeda for Takhzyro, a monoclonal antibody that treats hereditary angioedema, Daiichi Sankyo for Enhertu, which treats unresectable or metastatic HER2-positive breast cancer. We have 2 from Roche Genentech, Hemlibra and Polivy, the first being a subcutaneous treatment for hemophilia, and the latter being an antibody-drug conjugate for diffuse large B-cell lymphoma. And lastly, Portola Pharmaceuticals for Indexa, recombinant coagulation factor 10A. We're really pleased to have scientists involved with the first 5 of these products join us on our panel today. If you want more information on any of these products, there's a link available with 100-word descriptions of each product that you can view at the Galien website, including during this webinar. So what we'll cover today relates to this central question: can the foundational biological understandings now being brought to cancer be leveraged for targeted and/or personalized approaches to other conditions? So I'll start with a brief presentation to frame the topic. Then we'll spend the bulk of the time having the panelists describe their experiences in response to a few questions. And then we really want to hear from you, our audience. So we'll open it up for your questions, which you can type in for us to field. But before we begin, there's a brief survey. If you can put that up, please. The survey is being asked across all these webinars. Yep, it should be coming up now. Asking collectively what we think is the most important element of global policy to advance health innovations. So please answer. And the results will be shared at the end of this meeting, and also at the end of the week. All these webinars they're asking the same question, so you'll get an aggregated response across all of them. Okay, great. All right, let me jump into the presentation. So the starting point is this, which is Eroom's law. So what this shows is the change in productivity, R&D productivity for the biopharma industry over a series of 6 decades. And what it shows is an exponential decline in that productivity. The y-axis here is a log axis, so this is exponential decline. It's sort of a Moore's law in reverse, which is why the folks who published this in Nature Reviews of Drug Discovery back in 2012 called it Eroom's law. The good news is that since this was published, productivity has really turned around. And this is from a paper published this year in Nature Reviews of Drug Discovery called Breaking Eroom's Law, which shows that decline not only didn't continue, but actually reversed, and productivity has gone up in the last decade. And the question is why? And the answer is largely about better mechanistic understanding of disease. So the vanguard of this is in cancer, and you can see on the left actually a publication that was spearheaded by Desmond Hellman in the National Academies, showing the evolution of our understanding of non-small cell lung cancer as just one example, where we really moved from thinking about disease in terms of organ and histology to the mechanistic drivers. And on the right-hand side of this page, you can see the impact of that with mortality coming down. There are some social drivers of that mortality decline, including lower rates of smoking. But this is also largely about better care enabled by this better understanding of disease. We also see this outside of cancer, and this is from that same Breaking Eroom's Law publication from Nature Reviews of Drug Discovery this year, showing in the first column that the number of new medicines with supporting genetic data has been increasing. The number of new medicines approved at more precision levels, lower levels of indication in the ICD codes are increasing, and the number of new medicines that are approved for rare disease increasing, all of which are, typically are around monogenic disorders. And so we see this trend, particularly in cancer and rare disease. But those are probably the easiest areas because those are areas where you've got a tumor to type, or you've got a single gene disorder. The question is, how much can this be applied to more common chronic diseases, which often are polygenic? There are lots of genes involved, each of which have only a small impact. And so how do we tease out from that what's the right mechanism to go after? And so to answer that, we have a fabulous panel. So just to introduce all of them, the first is Beth Ann Collar, who is Executive Director of Global Clinical Development at Merck. Dr. Collar has her PhD in medical microbiology and formerly led preclinical virology at GSK and led R&D at Hawaii Biotech. Representing Sanofi, we have Bernard DeLay, Regulatory Therapeutic Area Lead. Dr. DeLay studied molecular biology at the University of Leuven and was head of regulatory affairs for Ablinks and involved in the development of Cablivi across all clinical phases. After Sanofi's acquisition of Ablinks in 2018, he has continued to support Cablivi. Representing Dompé, we have Flavio Mantelli, the Chief Medical Officer. Dr. Mantelli is an MD focused on ophthalmology, who also has a PhD in regeneration and has worked on nerve growth factor, including with oxybate. Representing Takeda, we have Dan Sexton, strategy lead for the Rare Disease Drug Discovery Unit. Dr. Sexton has his PhD in biochemistry from the University of Windsor and worked at Dyax for 2 decades, following it through to Shire and then Takeda as part of those mergers. And representing Daiichi Sankyo, we have Javad Shahidi, Vice President of Global Oncology R&D. Dr. Shahidi is the global team leader for Enhertu, on which he has worked since it was in phase 1 development. Dr. Shahidi has a passion for cancer research, which we hope to leverage in our discussion today. And actually, Dr. Shahidi or Javad, perhaps we can start with you, given your oncology background, but with a question that I think all of our panelists could weigh in on, which is, so we've seen great progress as just described. Why did your company choose to focus the way you did? And what lessons do you draw for, what might be applied for targeted therapies in other diseases?
Thanks, Michael, and thanks to the Galien Foundation for the opportunity. So there's a lot to talk about the progress in the past decades in oncology. But if I want to summarize in 2 or 3 minutes that I have in the opening statement, I think a lot of that stems from deep understanding of biology. You actually made a very good example in your opening statement about lung cancer. It wasn't long ago when lung cancer was just a single entity. And we thought we have a great progress when we moved to a histologically defined categorization of lung cancer. Actually, at that time, even treatment options were different between a small cell lung cancer, and choice of chemotherapy for squamous lung cancer might have been different from non-squamous. But we didn't stop there. And as you showed, lung cancer today probably is a collection of several distinct diseases. And the good news is that we're continuing to make progress even within lung cancer. Even in the past year or two, there are new targets that are showing as promising, at least have potential. And new approvals are coming based on new targets in lung cancer, further dividing this disease into actionable diseases with our new drugs. HER2 is one example that we are working on it as a potential promising next target. But I think importantly, the second part of this deep understanding of biology, what was combined with a deep understanding of drug candidates pharmacology. So as long as the understanding stops on understanding, you don't have a target to actually act against. The good thing about in the case of lung cancer, for example, is that that understanding led to development of actual drugs that can have actual benefit for patients with a disease that at some point, again, we knew it as one big entity as lung cancer. So I think that's the foundation of a lot of these progress. Another part, and maybe in the next part I can a little bit expand on it, is that the framework of clinical development in oncology also allows for these type of innovation. Perhaps we can talk about it, but I think that the bottom line of this is deep understanding of biology, deep understanding of pharmacology that speaks to that biology of the disease.
Great, thank you. Maybe Bernard, same question to you. How do you think about the progress that we've seen and what it might mean for a broader set of indications?
Well, if I look at Cablivy and acquired TTP, then I mean, for sure there is some genetic linkage with respect to risk factors, genetic risk factors for acquired TTP. For instance, prevalence is much larger in women versus men. Black people also seem to be more at risk for ATTP. And there have been studies that have shown that certain HLA alleles, human leukocyte antigens, increase the risk for ATTP. On the other hand, there are also alleles that seem to be protective. So there is definitely some genetic factors. You have to realize ATTP is an autoimmune disease, so it's also quite complex. There are definitely genetic factors that might determine the risk for acquiring the disease. But for the moment, it's less clear how these genetic risk factors can be applied in guiding treatment, for instance. Maybe later on I can expand a little bit more, but there are definitely a number of other markers like platelet count levels, like Adams TS13 enzyme activity levels, which is the enzyme that processes from Willebrand factor, and if the enzyme is impaired by autoantibodies, then you're at risk for the disease. So monitoring all these parameters can definitely and is used practically to guide treatment, to decide when certain modalities of the treatment should be initiated or intensified or stopped altogether. Yeah, actually genetic testing or anything that's at the moment not on the agenda. But of course, identifying and further studying those genetic factors may lead to a better understanding of the pathophysiology of the disease. And in that respect, it definitely would be useful to further modify treatment modalities for ATTP.
Yeah, let's come back to that theme around being able to dive into, using the genetic hits, not so much as personalized treatment, but as clues onto what's driving pathophysiology. I'd like to come back to that, but before we do that, maybe Flavio, if you could jump in again on the same question, sort of the general themes as you see them.
Yeah, thank you. I strongly agree with my colleagues about the strong need to get a deep understanding of the biology behind the diseases. It's not only about genetic diseases being monogenic or having a genetic predisposition. One example is the drug that the company for which I work had approved. That's not for a disease that is genetic per se. There are genetic conditions that can cause this disease, and in which replacing, as we did, a missing protein which is a biotech product, and it's totally innovative in its concept, can also approach the same problem from a different point of view. But what's important is that we have a very strong understanding of the mechanism of the disease, of the biology behind the disease. Then the shift that the pharma world has seen in the approach to the clinical development can answer in many different ways. There is not only genetic treatments, there are not only biotech products, The important thing is the approach where we start from understanding the biology of the condition, where we start from understanding sometimes what's missing, and we can act in the best way to answer in a context where, you know, all patients are different at the end, despite there can be some predisposing factor, there can be some aspects of the disease that can target the same way. Once we understand the biologic mechanism of the disease, we can certainly offer a better therapeutic option and an easier approach to clinical development that also results in faster times to get an answer to patients.
Very, very clear. And again, I think starting with the opening statement, your general philosophy is a good place to start. So let me ask the same question to Beth Ann, please.
Hi, thank you. Thank you for the question, Michael, and thank you to the Foundation for this opportunity. So I work in vaccines, and vaccines are just, a bit different in that, with vaccines, what we're trying to do is develop a product that will be effective for everyone. So our focus is slightly different. On the other hand, a fundamental understanding of the disease, as the other colleagues have already mentioned, and as you highlighted, Michael, is really what's driving, I think, innovation and improvement in vaccine development. And I think is really exemplified by what we're seeing now with the amazing worldwide efforts to develop vaccines for SARS-CoV-2, where just all of the knowledge that we have gained is being applied, and where understanding the fundamental issue is more linked to the pathogen rather than the human host. There certainly are host factors that come into play, and in particular for people who are not protected by a vaccine, where it may work in 90% of people, those 10% of people that the vaccine is not effective in, understanding that there's almost certainly host factors that are involved, and that's critically important to understand. But thinking about vaccines for infectious diseases, our focus is often on, first and foremost, understanding the pathogenicity of the actual pathogen, so that we understand how that pathogen causes diseases in human hosts, and how to target counteracting that, how to prevent that disease. It's interesting. It's also applicable to antiviral development, I think, because we're often targeting specific proteins or genes within the viral pathogen. So an antiprotease, for example, protease inhibitors or replicase inhibitors that are often very specific for the pathogen itself. When we're lucky, I think that we have tools, we have antivirals, or we have vaccines that can cross amongst pathogens of related viruses or related bacteria. And I think that that's a part of ongoing innovation, is leveraging our learnings from one pathogen and trying to apply it to others. I think it also applies to the use of technology platforms. So when you have a vaccine platform, for example, the platform that was used for Vivo is a recombinant vesicular stomatitis virus-based vaccine strategy, and that strategy is now being looked at for a variety of different pathogens, and particularly viral hemorrhagic fevers and including SARS-CoV-2. So it does all come back to a fundamental understanding so that you can target your efforts. I think in the past it was all done very empirically, a little bit of try everything. But as we understand better the pathogen, we understand the host immune response to that pathogen, we can really focus our efforts and hopefully come much more quickly to safe and effective products. And I think the whole world is hoping for that with the SARS-CoV-2.
For sure, but also just an incredible story with Ebola, which we're all very grateful for, and especially the people in countries that are most affected. Dan, last word to you. What is your kind of opening philosophy here?
Yeah, thanks, Michael, and thanks to the organizers for the opportunity to discuss this. Yeah, coming from the rare disease background, I think in some ways, rare diseases have always been sort of a precision-based medicine approach. And so I think that, perhaps starts with working directly with patient groups and listening to patients and understanding what the unmet needs are. And so, if you think about the rare disease patients and the challenges that they face, it can even start at the diagnostic level and helping and for different diseases, like for example, hereditary angioedema, the rare disease that Takhzyro targets, trying to improve that sort of diagnostic journey that they struggle with. And then really, again, it comes down to what the other panelists have said, really understanding the diseases, the pathophysiology, to really develop targeted therapies for rare diseases, is a huge challenge, and I think is really what determines the success of the products. And then other aspects, as we develop these targeted therapies, understanding more about the pathophysiology in the patients and this sort of concept of continual improvement. So as we learn more about the pathophysiology and what's going on in the patients, I think there's opportunities to develop even better therapies. And in some ways, Takhzyro, lanadelumab is a good example of that, in that we had identified and approved an approved molecule, ecallantide, that helped us sort of understand the pathophysiology and the target. And then, this then led to the generation of this, in a sense, a second generation product that we advanced. And then, sort of lastly, the last thing I wanted to mention is just understanding this, this really focus on pathophysiology, I think, can help us understand other diseases that are sort of adjacent to the targeted indication, and perhaps other therapies that can benefit these other patients as well. Yeah, thank you.
Yeah, thank you. I'll come back to this question of adjacent indications, because I think that's an interesting one. But maybe back to Bernard. So, you had mentioned in acquired TTP that yes, there are some genetic associations, including with race and gender, but it's not. We're not at the level of information where you could even think about any kind of personalized therapy, and that's probably, I think, it's going to be generally true of common chronic diseases, where you can run a GWAS and you maybe get Manhattan plot where you've got 100 hits, but all of them have prevalence of a couple of percent, and explain only a couple percent of the variability in either disease susceptibility or the natural history of the progression and so on. And so you teed up this idea that that information is probably more useful as a clue to where you look to understand what's the pathway involved and what might be an intervention point. I'd like to hear you talk about that, particularly actually in immunology, right, where I think it's been mooted, that the way we characterize the diseases right now, is probably each one of them is a heterogeneous assemblage of underlying mechanisms that might cut orthogonally across them. So whereas right now we've got, psoriatic arthritis and RA and ankylosing spondylitis and, UC and, pick your immunology disorder, it may actually be that there's a set of, mechanistic drivers that, account for some portion of each of those diseases. And, the right intervention would be obviously along that axis. How do you go from a GWAS that has lots of possible hits to a real idea around what you want to go after?
Well, maybe just first to correct, ATTP is not a chronic disease. It's a very acute disease, and it's actually usually it appears in an emergency setting. Patient becomes suddenly very sick, you need to make well to set a diagnosis. That's the first challenge very rapidly. And then you need to decide on treatment also very rapidly, and rapidly means in a matter of hours. So there's little opportunity to doing elaborate testing, etc. There's a number of clinical criteria that on the basis of which the diagnosis is made. But the most important one is measuring of the ADAMS TS13 enzyme activity, which is the underlying, that's been the, let's say, one of the major breakthroughs over the past years in identifying the pathophysiology of the disease, that it is caused by a defect in the enzyme that processes von Willebrand factor. And that defect can either be genetic. You have also a congenital form of TTP, which is CTTP, which is even much more rarer. And then you have the acquired form, which is caused by neutralizing autoantibodies that are inactivating the enzyme basically. And that's more prevalent, but still with an incidence of less than 10 per million. So it's really a very rare disease. And the identification of ADAMS TS13 as being at this, let's say the cause, the underlying cause of the disease, the effect in that enzyme, that's been a real breakthrough. Now, like I said, I mean, there is a number of risk factors that may predispose patients for getting the disease. But of course, I mean, even if those risk factors are present, it doesn't say that you will get ATTP in the next days or weeks or months or even years. You may never have get it. I mean, it's just an increased risk that you have. So the important thing is, once you have a patient, to be able to diagnose him or her and then to optimize the treatment. And currently, the treatment for ATTP is based on targeting three pathophysiological axes. On the one hand, you have a plasma exchange, which removes the autoantibodies, which removes the ultra-large von Willebrand factor that causes the platelet aggregation that is characteristic of the disease, and which brings in fresh enzyme. On the other hand, you have immunosuppressive therapy, another, let's say, more important breakthrough over the past years. Immunosuppressive therapy, which obviously gets attacked or is targeted at removing or stopping the autoantibody formation. And then the third pillar is Cablivi, where you prevent ultra-large von Willebrand factor for adhering to platelets, and in that respect, immediately stop the buildup of the microthrombi that cause the tissue ischemia that leads to the organ damage in this disease. It immediately stops it, and it sort of buys time for the patient to respond to the immunosuppressive therapy. Now, it is important to continuously monitor the patient, because every patient has a different response to those three aspects of therapy. Some patients respond very rapidly within a matter of days or a week at most. Others take many days or many weeks even, and are refractory to treatment. So it is important to tailor your treatment to the specific response that the patient exhibits, and that can be done, for instance, by on the one hand monitoring obviously platelet counts, which are a measure of the acute phase of the disease. On the other hand, monitoring also the ADAMS TS13 activity, because as long as that activity is suppressed, it indicates that the underlying autoimmune component of the disease is still active. And you can, for instance, not safely stop Cablivi, because in our studies we have shown that if you stop Cablivi treatment with underlying disease still present, then the patient is at risk for relapse very soon, within a matter of days actually after stopping Cablivi. So there's a number of factors that are monitored on a continuous basis, but to what extent these are genetically determined, or to what extent genetic testing could inform on an individual basis or guide on an individual basis the treatment that is needed and how the treatment should be optimized. For instance, intensivation or optimization of the immune suppressive therapy, that is currently absolutely not clear. So basically, treatment is relying on a number of markers which are easily measurable and which can be relatively easily interpreted. And directly applied on modifying the treatment. So that's where we are at least for ATTP at this moment.
Yeah, great. Thank you. I mean, that was really revealing. Maybe to shift gears a little bit, Flavio. So oxybate, as you said, right? It's not about the genetic cause of the disease, and in fact, even trauma can cause keratitis, which obviously isn't a genetic cause. But you certainly have, mechanistic thinking. You're focused on a downstream, driver of repair. How generalizable is that thinking to other indications that probably have, very heterogeneous upstream causes, but might have a downstream, intervention that focuses on the mechanism of repair? I mean, NASH might be an example that comes to mind, but love to hear your thoughts on what lessons we can take away from oxybate more broadly.
Yeah, thank you. That's a great question, and I think going a little bit to give you some background, oxybate is a recombinant human nerve growth factor eye drop treatment that's been developed for a rare disease of the cornea, and that is called neurotrophic keratitis, in which there is a damage to corneal nerves, and in turn there is a progressive damage to the cornea that goes from lethal epithelial damage and defect. It progresses towards corneal melting and perforations. It's a blinding disease, and it was an incurable treatment because all that could be done before oxybate was developed was to try to slow down progression or surgically to put a patch on the damaged cornea. And I think this can be translated as a concept to other diseases only if you think of the disease as a model to understand the biology behind what happens. So the cornea is very peculiar in not having blood vessel. It's avascular because it has to be transparent. Otherwise, you don't have vision. And so it's a very unique tissue in which you need corneal nerves, not only to do what nerves usually do, so provide sensitivity, for instance, but also to provide nourishment. So that's why it's called neurotrophic keratitis, because the trophic support is missing if you don't have corneal nerves. So once you understand this and you go towards the mechanism of the disease and the biology and what's missing, so there is a release of neurotrophins that is missing from these nerve endings that are damaged. And then you can treat the condition because you give an exogenous form of nerve growth factor that is normally present in the human body. If you think of this small indication is a rare ophthalmic condition, but if you think about it as a model of a disease where you understand the biology behind it and you can go directly at the basis of the problem. You don't want to treat the symptoms. You actually want to interfere with the problem and stop the detrimental loop and actually provide a permanent solution for the patient. I think that's how you can translate into other diseases. So beyond ophthalmology, of course, other diseases that could even be, genetic diseases. That's, I think, to answer your question, how I would translate it. Probably have the mindset to think of a disease as a model, a biologic model, where you can work to define a specific treatment, to develop a specific treatment, even something completely innovative, as we did in this case, because it's the first topical biologic product,
And maybe a quick follow-up. And I know you spent a career with nerve growth factor. What other indications might be next in terms of where you think of its application?
So NGF has been studied a lot in the past. You may be aware that it's a molecule that was discovered actually by an Italian scientist, Rita Levi-Montalcini, who was awarded the Nobel Prize in the 80s for this discovery. But for a very long time, it wasn't developed as a pharmaceutical product because it was used only in an academic setting as a molecule extracted from mice, from submaxillary glands of mice. And other companies, large pharmaceutical companies in the 90s got interested in HIV-related neuropathy, in Alzheimer's, in Parkinson's, in several neurologic and neurodegenerative diseases, and also produced another form of the recombinant human nerve growth factor that was, however, produced in eukaryotic cells. And that's less effective in terms of, production yields of the molecule itself. And what we did was to develop a technique to produce it in prokaryotic cells. So we have an NGF produced in E. coli that is more effective, and you can have yields that are sufficient for scale up and for commercial manufacture. And I think that's also part of the story and the discussion that we have today in this panel. Innovation is also about innovating the mechanism that are sometimes behind the production of pharmaceutical products. This molecule itself, NGF, has been discovered 60 years ago and has been used in academic setting for many decades. But until someone came up with a formulation and a process for production that is actually cost effective, then there was no product for the patient. In fact, this is the first ever application of an NGF that's been approved by EMA and FDA.
Yeah, maybe to pick up on the theme of innovation and production, both as you talk about in the scale up, being able to produce in prokaryotic cells, but also maybe to generalize that to innovation in modalities. Bernard, I wonder if you could tell us about nanodrugs and shark antibodies and camelid antibodies and why this kind of innovation is valuable and again where it might play more broadly.
Right. Well, nanodrugs is maybe a misnomer actually. I mean, they are called nanobodies. Nanodrugs are reminiscent of buckyballs and nanotubes and all that kind of products. So that has nothing to do with nanobodies. Nanobodies actually are a type of antibodies that are derived from single chain antibodies that are that have been identified in camelids. Camelids actually also in sharks. And the variable domain of those antibodies, which are fully functional, those variable domains we call nanobodies, and they can be tailored in all sorts of ways. So they can be humanized, but they also can be linked together. So you can link multiple specificities together to make multi-specific nanobodies. You could also increase their half-life extension, for instance, by linking them to a nanobody that targets human serum albumin, for instance, and then they adopt the half-life of serum albumin. But one key advantage is that they are they are very small. Around a single nanobody building block, if you wish, is around 15-16 kilodalton. So let's say about 10 times smaller than conventional antibodies, which makes that they can be used to target all sorts of epitopes that are very difficult to access by conventional antibodies. Another advantage is that they are quite stable. We usually manufacture them also in microbial systems. So Cablivi, for instance, is manufactured in E. coli. And most of the nanobodies that we're currently developing are manufactured in yeast cells, Pichia pastoris yeast cells. So they are quite stable as well, and they can be administered via different administration routes. So Cablivi is administered either IV or sub-Q, but we have also developed a nanobody that can be administered via inhalation, for instance, using a nebulizer, and that was developed for the treatment of RSV infection. So there is quite a plethora of applications, and actually it's a very broad technology platform that can be applied on a variety of therapeutic areas. So that's sort of the strength of the platform, and we are currently developing nanobodies in also for immuno-oncology, for immunoinflammation. Cablivi is a rare disease, rare blood disease product. It was the first nanobody ever to reach the clinic, actually, and sort of validated the nanobody platform. But so it's a very versatile technology platform, and yeah.
Yeah, and I mean, fascinating. One question that's been mooted about nanobodies is 15 kilodalton, still quite large relative to a small molecule, but question of would it be possible to go intracellular, and I wonder if you could talk a little bit about that, the platform prospects in that regard.
Intracellular would be would be difficult, I think. So, but yeah, I mean, there's different ways that the platform can be further developed. You could, for instance, also attach toxic cytotoxic compounds to it to target cancer cells. For instance, that's something that's being investigated.
And one of the other nominees is in fact an ADC, similar in kind to that idea.
Yeah. So that's definitely a potential development. Like I said, multi-specific nanobodies are also quite hot. We have, or we are developing a number of multi-specific nanobodies in multiple therapeutic areas. So yeah, I mean that's things which are ongoing for the moment.
Great, thank you. So Beth Ann, I guess you teed up a couple of themes. One, obviously, how much vaccines are on the mind of everyone in the world these days, but also how with vaccines you're essentially trying to do the reverse of personalized medicine, right? You want an intervention that works regardless of variability in the host, and ideally obviously variability in the pathogen. So can you talk to us about what are the difficulties, particularly on the latter point, as pathogens can evolve, right? I mean, that's sort of one of the key features of that I guess they share a little bit with cancer as well, and so can escape interventions, and how do you think through and there's also some heterogeneity in the in what's circulating, so that there might be multiple strains to address. How do you address that variability to come up with a vaccine that really works across all circumstances?
Yeah, thank you for that question, Michael. And that's a little bit of a tough one, because I think there have been different approaches taken for different pathogens, and some more successful than others. So just thinking about some of the pathogens that we haven't been successful in developing a vaccine to date, HIV and hepatitis C, of course, come to mind immediately. And a lot of that, we believe, is linked to the variability of the pathogen, the ability of the pathogen to change and to avoid whatever we are inducing. Thankfully, perhaps not all pathogens are quite as clever as HIV and HCV. And so, for some pathogens like measles, for example, live attenuated or yellow fever, for example, live attenuated vaccine developed for measles back, in the 60s and 70s, or for yellow fever back in the 30s, has, held up for decades, and are still safe and highly effective vaccines. So we're grateful for that. In the middle of the road there, you sort of have influenza, right? Which is, I think, a great example of how pragmatically we've dealt with things, but where there's so much opportunity for innovation to do better. Because we all know that, influenza varies. You have both, drift and shift. So you have, the slight variation year on year, our annual influenza vaccines, and then pandemic shifts where it's really, a new hemagglutinin or neuraminidase type coming in that requires something fundamentally different. But, the vaccines that we have are based on those hemagglutinin and neuraminidase, and they vary. And so there's been a long effort over decades now trying to develop, for example, a universal flu vaccine. I think we're not quite there yet, but as we gain more and more knowledge, we're getting closer and closer. So I think, it's applying the knowledge of the immune system. So I think there's been amazing work done in better understanding, what are epitopes that are recognized across all different influenza strains, for example, and developing monoclonal antibodies that become actually tools to inform. They are potentially therapeutics, but they're also tools to inform vaccine development. Because if you can, impact a pathogen, in this case influenza, with a monoclonal antibody in all different strains, then if you could develop a vaccine that induces that same kind of immune response in people, then you could perhaps have a broader impact and a more sustainable and sustained impact. So, I think the approaches that we've taken have certainly varied. Obviously, for example, with pneumococcal vaccines, there are 90, I think the last count that I'm aware of, 90 different pneumococcal strains that cause human disease. Current vaccine is the current licensed vaccine is a 13-valent. There are new vaccines, 15-valent, 20-valent, that are under evaluation, always looking to do better. And then there are non-conjugated vaccines, including one of ours that has a 23-valent. So the approaches really are really, again, sort of targeted to the pathogen. And, it's interesting. I've worked on dengue vaccines now for the better part of almost 30 years. And, there's 4 viruses that cause dengue, and that should not be such a huge problem. But in fact, because of the interactions between those viruses and, again, the impact of the host immune response, we're in a situation where there's really unique considerations. And, the belief is that a vaccine must be quadrivalent in order to be both safe and effective. So it is sort of personalized medicine when it comes to vaccines, because just the problems are different. For Ebola, I mean, I think, we were really blessed that the Public Health Agency of Canada had done just some tremendous work in evaluating a candidate Ebola Zaire vaccine. In fact, it was originally, a little bit the serendipity of science, right? It was originally developed as sort of an in vitro tool that the scientists at the Public Health Agency of Canada were using in vitro, maybe a little in vivo, and they started to notice, wow, this is really immunogenic, and wow, it actually protects small animal models. And so it sort of evolved into a vaccine candidate from a laboratory tool, and it turned out to be a really innovative and I think life-changing vaccine for people who are facing Ebola. It's challenging, and I think you have to keep your mind open to many different options. There may be different solutions for different issues.
Right, you have a palette of ways to address it. Great, super answer. Maybe Dan, is shifting gears a little bit, but so hereditary angioedema, potentially life-threatening genetic disorder, as you described, right? You can you start by knowing the gene, but just knowing the gene doesn't get you a treatment, right? So how do you go from understanding that to what's the right intervention, what's the human biology that you want to affect, and what's the the treatment you want to develop?
Yeah, thanks for that question. So in hereditary angioedema, there's the main form is due to a genetic deficiency in a protein called C1 inhibitor, which normally controls a serine protease called plasma kallikrein. And so that was pretty well understood. I would say, and this was that concept of incremental innovation, perhaps. And prior to the development of Takhzyro, protein replacement had been used in these patients, and they responded well to that. However, as we got to know the patients and their unmet needs, there was clearly an opportunity for better, more efficacious therapies, and so one of the earlier treatments were sort of trying to target and trying to treat the attack as they occurred. And so, for instance, we have a therapy called ecallantide or Kalbitor that treats the attack, and that is also a plasma kallikrein inhibitor. And so that really, in a lot of ways, validated the target for those patients. But then there was still an unmet need in that, this was a therapy that was treating the attack after it happened. And so that's why, the idea of developing a monoclonal antibody with a long half-life that they could take prophylactically to prevent the attack, was much, preferred for those patients. And then the other thing I wanted to mention a little bit about, if I can go on a little bit more, is there's other types of angioedema and other indications perhaps. And, so and this is the idea of learning more about the pharmacology and pathophysiology. There's other patients with angioedema, for instance, that don't have that mutation, but there's more and more mutations being identified. I think now we're up to 5 or 6 different mutations. So it's sort of, I think, but they still have the same kind of disease symptoms. So I think it's building up the story more and involving more genes and so also implicating the fact that kallikrein is involved in those other adjacent indications. And the thing I didn't mention is that when kallikrein is overactive, like in these patients, it generates a propeptide called bradykinin. And so there's a number of indications, of course, that an excess of bradykinin could be involved in. And so you may have seen, even in some of the popular press lately, there's this concept of a bradykinin storm with COVID-19 associated pneumonia or acute respiratory distress syndrome. And so there's potential that targeting this pathway could be effective or beneficial, at least in those patients as well.
Yeah, that's I did not know that, but that is like you say, very interesting line of follow-up scientific inquiry. One last moderated question, and then we'll open it up for audience Q&A. And so I'll invite folks even now to start populating that. I think there's a Q&A button that you can press to put a question in. But last moderated question, Javad, you kicked us off. Maybe I'll ask you to close us. So oncology, speed is critical in every indication. And oncology has been one of the areas where we've been able to jump very quickly from phase 1 to phase 3, and obviously get medicines to patients that much more quickly as a result. Can you talk about the development frameworks that empower this kind of speed in oncology?
Sure, and it's fascinating to listen to other colleagues talking about it, because a lot of concepts are actually shared across therapeutic areas. But let me use the example of Enhertu and then apply some of these concepts. It took 4.5 years, from less than actually 4.5 years from the first patient dose in any clinical setting with Enhertu to the first approval of the drug. That's for biologic, that's pretty fast in oncology. And for an ADC, I would say with all the complexities around manufacturing, it was quite an achievement. But let's apply some of the concepts of why these type of speed and quality and innovation are possible, for example, in oncology. So biology of HER2, so this is a HER2 antibody drug conjugate. So biology of HER2 is, we're building on a heritage of 2 to 3 decades. We understand the biology. The ADCs, the concept of ADC is also not new. It's at least decades old. What our Japanese scientists did basically to systematically improve on the idea and build a structure that can efficiently deliver a potent cytotoxic to cancer cells with minimizing the side effects. That was basically what it is, and the attributes, improved attributes of any common ADC. Unmet need that Dan mentioned is an important. I think we do a good job in oncology in defining the unmet need and then pursuing our drugs in an area that we think speaks to an unmet need. So that I want to emphasize that was HER2 positive after several lines of treatment. So the median lines of prior treatment in our study was 6 for HER2 positive disease. So with little options, if you bring a deep response and quality of life, you are addressing an unmet need. So that's another thing. We didn't much discuss, but I think CDX and biomarker is a key component of this framework. We have to be able to identify these patients who are having a condition and are more likely to benefit from a specific targeted therapy. I think that's the key that is also, again, we're building on the heritage. We're not necessarily introducing a new CDX here. But I think one thing that I want to emphasize and wasn't mentioned is the endpoints. Relevant endpoints that allows us to measure the success of a drug. I mean, in oncology, the concept of surrogate endpoints, by the way, are not new. Some of these endpoints actually will lead to regulatory approval. But what I want to mention even further than that is that when you have an endpoint that speaks to your drug's biology, regardless of whether it ends up being the part of your regulatory approval on its own, it allows you for smart investment early on and rapidly identifying very promising drugs. On the flip side, also rapidly identifying the drugs that won't work. I mean, in case of ADC, for example, if you do all of the manufacturing scale up sequentially to your OS endpoint at the end of a phase 3, you're going to end up with a clinical development plan that takes decades to bring a drug to the market. But with an endpoint like response rate, where relevant and where applicable, with a duration of response, you can make these decisions sometimes on 20 or 30 patients with deep response, identify that this drug is different, this is promising, and I am ready to invest hundreds of millions of dollars for manufacturing so that by the time my clinical package is ready for submission, I also have, for example, in this case, my manufacturing scaled up and ready to go. I think all of these components come into play, and I think in oncology, you have several examples of rapid development and rapid submission and approval. And I think they all use the similar concepts, and all of them match and lead to the final results that is, in this case, access of a very promising drug to patients with a lot of unmet need.
Yeah, it's a great example. You know, good drugs declare themselves early, often in a very small patient population. I'm checking the Q&A, so it doesn't look like any have been submitted yet. So really encourage the audience. This is a unique opportunity to, you know, ask either general questions or specific, you know, around the drug and indication in question. But while we're waiting for those, maybe I'll I'll pepper you all with another question that's on my mind, which is, this industry has been, somewhat unfairly, seen negatively by the public. And I think that's partly through lack of understanding of just how much goes into the innovation process. Can you, this is an open question, so, folks, more than one to jump in would be fine, but, can you talk about some of the challenges that you faced along the drug development process and things that might be, if they were shared and sort of the public were more aware of just how difficult it is, might bring, some recognition of the kind of, work that goes into this process. Would love to hear. I'm sure you have plenty of challenging situations that you can bring to mind from the process. To anyone on the panel who wants to jump in.
And if I can, yeah, about the ATTP, I mean, one of the challenges obviously is the rarity of the disease. I mean, I don't have to tell you how difficult it is to conduct a trial in a disease that has a prevalence of yeah, let's say a few patients per million inhabitants. So finding the patients already is a challenge in itself. Just to give an example, our phase 2 study, or first study in patients actually took about 3 years to complete. We initially anticipated recruiting 110 patients, and then seeing that recruitment went really very slow, we stopped at 75. But fortunately, we met the primary endpoint, even with less patients than expected, and that gave us the signal to continue the development and then do the phase 3 study, which was recruiting a bit or significantly better, because by the time we had publications out and let's say more confidence in the medical community about the efficacy of the drug, basically. So rarity, I think, is at least in our case a main issue or a main limitation. The other limitation is the fact that the disease is an acute disease and occurs in an emergency setting. So patients come in, need to be diagnosed very quickly, and the diagnosis itself is also something that has been shown to be a limiting factor. Sometimes it takes quite a while, especially in centers which are not experienced with the disease, because it's so rare. It can take days before the patient is diagnosed correctly, and that's every day is a day lost, and may lead to the may increase the mortality, and the patient may die before he's actually properly treated. And the rare disease context and the emergency setting also make it more difficult to conduct operationally the clinical trial, for instance, to get informed consent for recruiting patients. So and then if you, in a broader sense, the fact that patients are so few makes it also more difficult to establish a suitable knowledge base on the disease. And for instance, if you would look at genetic factors, finding links between prognostic factors, how the patient will respond to the disease, etc., and genetic markers makes it all more difficult if you have less data to rely on. So I mean, that's at least for ATTP a number of limitations.
And Michael, maybe I can build off of what both Bernard and Javad commented. And in terms of clinical trials, just thinking about our Ebola experience, where it was an emerging, epidemic disease that comes, and generally the outbreaks are fairly short-lived. So being ready to conduct a clinical trial is not necessarily straightforward. And in fact, it was the World Health Organization's really innovative trial design where they used ring vaccination, a strategy that had been used to help eradicate smallpox. So it really focused the vaccine evaluation effort on populations that were most at risk because they were contacts of active Ebola virus disease cases. So where the other designs that were more traditional actually never captured any cases, the ring vaccination approach did. So sort of innovation in trial design to try to get in and get to the right population and get there quickly, as Bernard was highlighting. And then thinking about manufacturing, and just want to, highlight, I think, for a vaccine, especially, a live virus vaccine, even non-replicating vaccines, these are complex biologic systems that take a long time to manufacture. In fact, very extensive testing. So, from start to finish, from pulling the first vial for Ebola vaccine, for example, it's about a year until you have released product. So it takes time. It takes time to build the facilities, to validate the facilities, to validate those processes. And as Javad was highlighting, coming quickly, from the first patient in for the Ebola vaccine, it was 5 years until the first licensure, and that was really primarily driven by manufacturing. That was the piece that took longer. The clinical trials actually happened very quickly, but we had to do the manufacturing piece, and that is the piece that I think people don't understand. And because it's sort of insider knowledge. Most people don't. It doesn't matter to them how we manufacture, but it has to be done the right way and with a very high quality, especially for vaccines. Vaccines are given to healthy people. So the bar is exceptionally high, and it takes time, and it's definitely a challenge. And, again, coming back that we're living in this COVID-19 era, the efforts that are ongoing to manufacture to try to vaccinate billions of people, it just completely boggles the mind. And we're, leveraging existing technologies that have been proven before and that can be scaled up rapidly is just so critical.
Great. Okay. Well, we have a few. I was pausing just to see if anyone else wanted to tell their tale of difficulty, but we do have a few questions that have come in, and actually the first one is back to you, Beth Ann. Which is that we've seen an uptick in productivity in drugs. The chart that I actually showed earlier, that was not about vaccines per se. The question is, are we seeing an uptick in productivity in how difficult or easy it is to create vaccines, and how likely are some of the new approaches in vaccines, such as VLPs, likely to bear fruit?
Yeah. So I mean, it's another great question, and I hate to prognosticate. I have to say. I mean, I think VLPs work really well for some targets. For example, for human papillomavirus, we have highly effective vaccines that are based on VLPs. Hepatitis B, for example, as well. But for other targets, they haven't worked so well. So it's really difficult to, I think, come up with a one-size-fits-all for vaccines. It really does need to be somewhere targeted for the specific product. And like with drugs, there's lots of things that we try that don't work. So I don't know that I would characterize that as, lack of productivity, but it takes time. And sometimes we have good animal models, for example, that allow us to do some early triaging of approaches, but not always. So, yeah, I mean, there's certainly a lot of work ongoing. And I actually do think that between the Ebola experience that already, I think, demonstrated ways to think creatively and to move more quickly, and now COVID-19, that I think it's triggering, huge amounts of innovation and creative thinking, and hopefully will lead to a lot of new activity in the vaccine space. Great,
thank you. Okay, and now a policy one, maybe to the full panel. And one that the question asker says, perhaps this is a little controversial, which I think it may be, but I don't think we shy away from controversy, which is, why doesn't the industry invest more in education around what it takes? So K-12, films, and other ways to shift cultural norms. anti-science sentiment is a large societal problem. The pharma industry could help, actually, particularly around vaccines. Obviously there's current question around how many people would even be willing to take a COVID-19 vaccine, but I think not just vaccine, right? I think there's a general question around people understanding what it takes in the industry. So what do folks think about that in terms of investing in educating broader public, but also in school setting?
So if I can start and then ask other colleagues to comment. So I think by the way, it's a big item of science education. It certainly goes beyond pharmaceutical industry. I think I agree with the tone of the question, that can we do more? I think that's certainly something to consider. The realities are that sometimes we're also limited by the rules and regulations and where we can go and how we can go and what we can say. I think that's an important aspect of it. Again, I don't think it stops us from further contributing to this overall concept, but if I can call it the concept of science literacy, I think it certainly goes beyond pharmaceutical industry. It goes beyond medicine, and it is something that is more cultural and mindset type issue that societies as a whole should address. And I absolutely agree with the person who is asking the question, that perhaps pharmaceutical industry can play a more active role in it, but it certainly is much more multifaceted than the work of one single industry.
I fully agree to that. Actually, I picked more public trust in science as the fifth poll question, as one of the most important things to focus on. Yeah, and indeed, it's all linked to education and people understanding basic scientific principles, because that's where it goes wrong, I think. People just don't understand what science is about and how science works, and so making people aware of that, and I think is primarily is a major task. The only skepsis I have around, I mean, I fully agree that pharmaceutical industry should play their part in that. The only skepsis I have is that everything that comes from the pharmaceutical industry will also be looked at with suspicion from a certain fraction of the population. It's like the tobacco industry educating people around the dangers or not of smoking. So I mean, whether what they say is true or not, people will look at it, hey, they are an involved party and they have an interest in whatever they sell, and yeah, what they tell will be colored in a sense.
Yeah.
But the basic message I fully agree with. I mean, there is definitely a big task in educating the wider population.
Great, and actually the fact that you mentioned your poll response is a great segue. If we can call up, we just have a minute or two left, so if we can just call up the responses on the poll. No, not too many votes. I guess your vote didn't go through.
2 votes.
You voted for number 5, but definitely universal access to medicines and vaccines was the winner with the N of 2. So let's not interpret it too much. But we will have an aggregated response across all the panels at the end of the week. So quite interested in that. I want to thank all our panelists for a really fantastic panel. The webcast from today is viewable and shareable with others. All you have to do is go to the Galien Foundation homepage. We look forward to seeing you at our other webinars as well, and our Thursday award cocktail and presentation of the 2020 Vagilos Pro Bono Humanum Award winner, Dr. William Fagey. So thank you all and see you soon. I think we have a little video on the way out, but again, really appreciate all the effort from our panelists. It was great.
Best biotechnology product.